The procedure presented here shows an easy method of preparing mica-supported lipid bilayers suitable for high-resolution imaging such as TIRF single-molecule tracking studies. The mica surface is prepared by splitting off a sandwich of a single sheet of mica glued between the glass microscope slide and the cover slip using optical adhesive. That exposed a flat and extremely thin fresh mica surface. By mounting a small plastic chamber made from a 1.5 ml vial cap, the volume of lipid vesicles required for bilayer formation can be as low as 30 microliters. As a first step, take the multi-layered sheet of mica as it comes from the supplier and cut it with scissors into smaller pieces. Cut mica leaflet into 2 or 3 thinner leaflets using a fresh knife blade. Now, clean the cover slips thoroughly. This involves placing 0.17 mm cover slips into a glass staining jar, each cover slip in a separate slot, and sonicating in detergent for 30 minutes at 60 Celsius degrees.